This is a production of Cornell University.
All right, so today I'm going to be talking to you about two different parts of development. So I'm going to talk about things happening in the flower and things happening in root hairs. So that hasn't made it onto our website yet, but we've been thinking about root hairs a lot in the past couple of years. So first I want to talk, so what you'll notice is when I have a flower up here, that means I'm talking about reproduction, and when there's a root, I'm talking about below ground stuff. So my first love is communication in plant reproduction. And probably I don't need to tell most of you here, but what happens is we have pollen lands on a stigma. There's communication between the pollen and the papilla cell. If it's a compatible interaction, a tip-growing pollen tube will grow, and then it needs to make its way to the ovules, which contain the female gametophytes. And during this process, there's a lot of intercellular communication occurring as this pollen tube encounters various different cell types. I think it's 6 or 7 different types of cells. The pollen, its whole purpose in life is to carry the 2 sperm cells to the female gametophyte so that double fertilization can occur to make the embryo and the endosperm. And the female gametophyte also has these specialized cells called synergids. And synergids are my favorite cells. So they're haploid cells. They're not gametes, but they're in charge of telling the pollen tube what to do when it reaches its target. Okay, so we basically have 2 highly polarized cells that need to talk to each other during pollen tube reception. And so we have tip-growing pollen tubes that grow by adding material, new cell wall material and new membrane only to the tip, so they don't get wider, but they get longer. And then inside ovules, we have these synergid cells that are also highly polarized cells. Here's the stick. And we can see here the 2 synergids. Here's the egg and the central cell. And these synergids have a highly specialized region called the filiform apparatus. So this filiform apparatus, so this is a scanning block-based SEM through an ovule, showing the 2 synergids. Here's the egg, central cells back here. And we can see there's dark material here. This is a membrane-rich region. And if we segment this, we can see that it looks like fingers that project up into the cytoplasm of the synergid cell. And this is where all the action takes place in the communication between the synergid cell and the arriving pollen tube. And we know that communication is very important from some studies that were done by Higashiyama and Hamamura back in 2008, where they took an ovule of Torrenia and placed it on pollen tube growth media. And then they germinated, they had a pistil over there and let pollen tubes come out. And what the scientist is doing is moving the ovule away. And what I hope you can notice is the pollen tube is following the ovule. And this suggests that the ovule, specifically these 2 synergid cells, are emitting molecules that then the pollen tube has a receptor for, and the pollen tube can change its growth direction in response to signals from the female gametophyte. Higashiyama's lab identified these signals, and they're actually small cysteine-rich peptides that they named LURs. And then the pollen tube receptors for these LURs have also been found. So that's first indication that synergids are really important for telling the pollen tube what to do. Second, in this study, this was done in Ule Grossenklaus's lab by Queen Yo, who was another postdoc when I was there. And what we have here are 2 synergids expressing calcium reporters, and then we have a arriving pollen tube. This is a minute timer. And I hope you can see, and red means high calcium, blue is low. As the pollen tube starts to talk to the synergids, we get these calcium oscillations happening in the 2 synergid cells. Also, calcium levels increase greatly in the tip of the pollen tube. And in this case, after about 40 minutes, the pollen tube then starts to grow again. And it gets up near the top. and burst to release the sperm cells. The second synergid stays alive, and it acts as a backup. So it can attract a second pollen tube by sending out lures again to get another pollen tube to come if the two sperm cells from the first one can't fuse. So then the same process can repeat itself. And recently a group in China showed if both tries don't work, then the central cell can also send out a signal and attract another pollen tube to try yet again. So the plant really wants pollination double fertilization to occur. Okay. So when I was a postdoc in the Grosvenor lab, we were taking a genetic approach to try to understand the molecular recognition of pollen tube synergid communication, which we call pollen tube reception. And so mutants on the female side that disrupt this process, they can still attract a pollen tube, but the pollen tube doesn't burst. So it doesn't get the signal, it's okay to release the sperm cells. Instead, it keeps growing around and around. So the first mutant discovered was Feronia, and this encodes a receptor-like kinase. It's expressed in synergids, but it's also expressed throughout the plant in most cells except for pollen. But its gametophytic phenotype is that if you have a heterozygous Feronia mutant, half of the ovules will remain unfertilized because the pollen tube doesn't get the signal to burst. When I was a postdoc, I was working on a second mutant, which we named Nordia. These are named after Etruscan goddesses of fertility. That's Uli, not me, naming them. But this one was really interesting. So similar phenotype, pollen tube overgrowth, pollen tube doesn't get the signal to burst. In some ovules, the pollen tube can burst. So it's not an all or none like the Feronia phenotype. And so we see lower levels of infertility than in Feronia. It's also shown in Uli's lab in this study that those calcium oscillations, if you quantify them, we can see in wild type, we can see in both the receptive synergid and the non-receptive synergids, we have these calcium oscillations. And we also have it in the pollen tube as well. And then after the pollen tube bursts, we see the pollen tube's dead now, and then also the receptive synergid, the receptive synergid will die, and then the other synergid stays alive. In Feronia mutants, those oscillations don't occur at all in either the receptive or non-receptive synergid. But in Nordia mutants, they occur, but they're at a lower amplitude. So the idea was that the reason why Nordia mutants aren't an all or none phenotype is that sometimes stochastically you get enough calcium oscillation that then the pollen tube gets triggered to burst. Okay, so from all this and from work that several labs have done, there's a model that we have Feronia and a GPI anchored protein called Lorelei that are found at this filiform apparatus, and they act as a co-receptor for Ralph peptides from the arriving pollen tube. And this co-receptor then triggers a signal transduction cascade, which leads to calcium oscillations in the synergid. And then I'm going to talk a little bit more about this Nordia NTA and what happens to it during this process. Okay, so how does Nordia control the amplitude of synergid calcium oscillations? So Nordia is actually an MLO protein. This stands for mildew resistance locus O. The Arabidopsis genome has 15 members of this family, and all of them are predicted to have 7 transmembrane domains and a big extracellular loop. There's 2 cysteines that make a disulfide, predicted disulfide bond here, and then this cysteine and this cysteine make another disulfide bond. In the C-terminal tail, there's a calmodulin binding domain. MLO proteins have been popping up a lot lately. So they were originally discovered in the context of powdery mildew infection. So they were discovered in barley, and if you have a barley MLO mutant, the plant becomes completely resistant to powdery mildew infection. Later, it was found that clade 5 MLOs in Arabidopsis play the same role. So a triple mutant becomes completely resistant. Single mutants have lesser degrees of resistance. Nordia is involved in pollen tube reception. And then there's another 4 MLOs, 1, 5, 9, and 15. If you knock all of them out, the pollen tubes burst early, so they can't maintain their integrity. The MLO disease phenotypes are conserved across monocots and dicots. So basically, any plant that gets infected by powdery mildew, if you can find the right MLO and knock it out, then the plant becomes resistant. So this means that we don't know what those MLOs are actually doing for the plant, but the powdery mildew takes advantage of them. We also showed back when I was a postdoc in collaboration with Ralph Panstruga's lab that Feronia mutants, so the receptor mutants, are also resistant to powdery mildew. So this gave us this idea that there's some conserved pathways. And so we have powdery mildew, or a tip growing cell that must communicate with and penetrate, make a interaction with a plant cell. Pollen tubes are also tip growing cells. They must communicate with the female gametophyte to deliver the sperm cells. And in both cases, it was shown that MLO proteins actually accumulate, so they become polarly localized to the site of tip growing cell communication. And Nordia also does this. So before pollen tube arrival, Nordia is localized in the Golgi, and then when a pollen tube arrives, there's signaling that moves it to that filiform apparatus. And we can see that here. So these are synergid cells. Before a pollen tube arrives, we see these punctate pattern with complete overlap with Golgi. And then when a pollen tube arrives, we can see this starts to accumulate at that filiform apparatus. Okay, so we wanted to take a closer look at this accumulation. So what we used was a semi-in vivo pollen tube guidance and reception assay that had been developed by Leonor Bovita and Ravi Palnavelu and several other groups in the plant reproduction field. And the idea is that you can germinate pollen on media and it'll make a pollen tube, but those pollen tubes can't be attracted to ovules. So what we need to do is to apply pollen to a stigma, give it a few hours to grow through, and then we cut the stigma. We can put it on media and arrange ovules around, so then we can follow what happens to the pollen tubes and what happens to our Nordia protein during pollen tube reception. So here's a live movie of this, and if you watch this ovule right here, we can see when the pollen tube approaches, we see a shift in the GFP signals. So that was exciting, but we didn't know if that was a cause or a consequence. And so Jing, my former PhD student, did a lot of live imaging. So we have a DS red labeled pollen tube, and then we have Nordia GFP. And I hope you can be convinced that we see a shift in the signal. We can look at time here, and we see that this actually happens, starts to happen as soon as the pollen tube makes contact. It arrives at the synergids. At this point, we wanted to test whether this was required for pollen tube reception. And so we took advantage of an earlier study we had done where we expressed different MLOs under synergid-specific promoters to see if they could complement the Nordia phenotype. And we came upon one of the MLOs, MLO1, that actually constitutively goes to the when expressed in synergids. This one does not complement the Nordia phenotype. But we thought we could take advantage of this to try to do some domain swaps to manipulate Nordia accumulation patterns. And I should also mention in epidermal cells, so this is in tobacco transient expression, Nordia goes to Golgi and MLO1 goes to the cousin membrane. So we did a bunch of domain swaps and we found that the C-terminal cytoplasmic tail was sufficient to make Nordia, a Nordia chimeric protein, accumulate mostly at the filiform apparatus. And then we tested whether this could complement the Nordia mutant phenotype, and it complements just fine. So I'm going to show a lot of these graphs. We have percent unfertilized ovules. Sometimes it will be percent ovules with pollen tube overgrowth. And we can see in Nordia in this experiment, it was about 40-some%. And our Nordia MLO1 was able to complement. So that meant it wasn't dangerous to have it there early. One hypothesis was maybe it's poisonous and it needs to stay out of that region until it's necessary. That did not seem to be the case. Okay. We also knew that Feronia itself is required for this trafficking of Nordia to the filiform apparatus. So in Feronia mutants, Nordia stays in the Golgi, it doesn't go to So the obvious question was, can we take our constitutively filiform apparatus localized Nordia, which we now call FA Nordia, could that complement the Feronia phenotype? So we did this experiment. So that's FA Nordia, this is regular Nordia, and we put it in a Feronia background, which has 80 to 90% infertility. And we can see, we start to see seeds developing. We can quantify that. So Feronia here, this is a homozygous Feronia mutant with 80% infertility. We can complement the phenotype to about half using our FA Nordia. So, and we can also check pollen tube overgrowth, it's the same thing. So yeah, we can actually bypass a very important signal transduction pathway by just moving a protein to the place in the cell where it needs to do its action. And also we showed that Feronia is not necessary for Nordia to perform its function, or the MLO to perform its function. So at this point, we still didn't know what MLOs did molecularly, but we came up with what we called our booster signal hypothesis. And that was that Feronia was absolutely required to give a signal that would lead to pollen tube bursting. But also it causes trafficking of Nordia to this filiform apparatus, which then helps to boost the signal to make sure that the pollen tubes will burst. So if we take out Feronia and just have a Nordia mutant, and just have Nordia filiform apparatus, the FA Nordia, it's able to do the function. So that was our booster signal. And somehow we also think that Feronia, either by increasing calcium, leads to this trafficking, or it's directly somehow intersecting and causing Nordia trafficking. Okay, so the question was, what are signals X and Y from Feronia and Nordia? Are they the same molecule? And for this, we and others went back to this observation that in Nordia mutants, the calcium oscillations occur at lower intensity. And Sheng Luan's group in 2022 tested the hypothesis that maybe MLOs are actually calcium channels. And so they took all 15 Arabidopsis MLOs, expressed them in COS7 animal cells, and they found some of them could conduct calcium, but Nordia couldn't, and MLO1 couldn't. Then they took advantage, then they took a closer look at where these proteins were going in the COS7 cells. And Nordia GFP actually looks in a punctate pattern very similar to Golgi. But if they co-expressed with Feronia and Lorelei, the protein would go to the plasma membrane. Or if they used our FA Nordia construct, the protein would start to also appear at the plasma membrane. So that's it. And then they tested whether the plasma membrane localization of Nordia or FA Nordia would lead to calcium intake into the cells, and indeed it did. So the conclusion was MLOs or maybe calcium channels, they need to be at the plasma membrane to do their job. Okay, so now we know Nordia acts downstream of Feronia. It regulates calcium influx into the synergids. Another question we're trying to answer in my lab is, how do those calcium oscillations in the synergids lead to pollen tube rupture? Stay tuned, that's a really hard question to answer. But we think it might actually be related to not what's going on in this cell, but what's actually going on outside. So maybe we're depleting calcium outside the cells, and that's allowing cell wall changes to occur that are then going to let that pollen tube burst. Okay, now I'm going to change gears a little bit. We're going to go underground. So how many people in this room had heard of Feronia before I mentioned it in this talk? Yeah, it's pretty famous now. Its first publication was that it's involved in pollen tube reception. And since then, it's been implicated in a lot of different things. So immunity, not just the powdery mildew, but also bacterial immunity, cell growth, stress responses, morphogenesis, cell wall mechanical integrity. It's been proposed to be a cell wall sensor. And so we made a hypothesis that maybe there was a conserved signaling module where Feronia activates an MLO downstream, and this helps cells to communicate with their external environments. So what we did is we took our FA Nordia GFP, which is a gain of function channel, and we expressed it this time with the Feronia promoter, so it would be expressed everywhere where Feronia would be expressed, and also with the 35S strong promoter that would be active in sporophytic cells. And then we tested what phenotypes of Feronia could we complement with FA Nordia. And so first we looked at just our pollen tube reception phenotype that we had studied using synergid-specific promoter MIB98. And we found that both the Feronia promoter-driven FA Nordia could complement it at about the same level as the MIB98. But surprisingly, the 35S promoter construct could complement completely. And the lore had been that 35S is not expressed in mutaphytic cells. It actually is sometimes. So not all the time it's a positional effect, but 35S can be active in synergids. A second phenotype that we tested, so it had been shown that so when pollen tubes leave the transmitting tract, usually only one leaves at a time. And this is, there's a Feronia mediated gating mechanism here that sort of opens the gate, lets one pollen tube out, and then recloses it. And the phenotype in Feronia plants is that more than one pollen tube will leave the transmitting tract at the same time. Sorry, this is getting cut off. That one's fine. And indeed, this was with an interspecific cross. It also happens with conspecific crosses. Our FA Nordia could also complement that. So just having an activated MLO channel is enough. Interspecific crosses. So if you cross Arabidopsis thaliana with Arabidopsis lyrata, one phenotype is so pollen can germinate on a stigma, send a pollen tube through, but in a lot of ovules, the pollen tube doesn't get the same burst. And if we express FA Nordia or if we overexpress Nordia itself, we can actually improve interspecific pollen tube bursting in these interspecific crosses. Interestingly, in a Feronia background, this doesn't work anymore. So we need Feronia for FA Nordia to be able to do its job in these interspecific crosses, which is still a mystery. Okay. Finally, a lot of people have looked at Feronia root phenotypes. So Feronia roots actually are longer than wild type roots. This is all in Arabidopsis thaliana. But the more striking phenotype is that Feronia root hairs are stunted and they often burst early, so they can't maintain their tip integrity. That phenotype is completely complemented by expressing FA Nordia with either the 35S promoter or with the Feronia promoter. I'm only showing the 35S data here. We took a closer look. Oh, sorry, this looks bad. We took a closer look at Feronia root hairs. And what we saw was that not only do they burst early, they tend to be more triangular shaped. And also, we also sometimes see spherical root hairs. So they've completely lost their polarity. And those phenotypes can all be complemented by having our FA Nordia construct. Also, if we add FA Nordia, the root hairs become more less triangular in shape. So this is measuring width over base, tip width over base width, and we can see we go back to. Finally, another phenotype of Feronia is that the plants are dwarfed. And that phenotype could not be by either Feronia-driven FA Nordia or So from this, we conclude that some, but not all, Feronia phenotypes can be complemented. So pollen tube overgrowth, polytubie from the septum, primary root length, abnormal root hair growth, but interspecific pollen tube reception and vegetative dwarfism cannot. So what we think is this Swiss Army knife Feronia, some of the things it does need calcium downstream and some don't. And so the ones that need calcium, we can bypass with an MLO. Okay, so what about calcium? So I told you MLOs are thought to be calcium channels. We know from lots of groups' work that there's a tip-focused calcium gradient that's established after root hair initiation, and it's linked to polarized root hairs. I showed you earlier that we have this calcium gradient or calcium oscillations that occur in synergids upon pollen tube arrival. And that this is also linked to being able to have a pollen tube that bursts and releases its sperm cells. So we decided to take a look at what happens to calcium in Feronia and F. nordia root hairs. So for this, we used an R-gecko marker just because we had it in the lab. We understand that chameleon is better. But we used light sheet imaging to capture images every 3 seconds for 12 minutes from growing root hairs. And then we measured what was happening with calcium at the tip of the root hair. Here's the light sheet system. So this allows us to do sort of 3D imaging in time. And here's what we see, and I'm going to need to do this. So in Colombia, you can see these calcium oscillations in root hairs. In Feronia, we can see round spherical root hairs. We can see some root hairs trying to grow. And then these flashes are root hairs actually bursting. And when we add F. nordia back in, we now recover tip growth and we see oscillations occurring again. We also quantified this. And again, I'm sorry about the quality of that picture. I didn't realize it did this. But what we see is in Colombia, if we just look at the oscillations, this is from one example, versus Feronia, we see in Feronia, they get very erratic. So they're kind of out of control. In the spherical root hairs, we see very little calcium. But if we add back in 35S F. nordia, we can restore the calcium oscillations pretty much to normal. So that's good, right? So maybe we're complementing Feronia root hair phenotypes by restoring the ability to do calcium oscillation. So that F. nordia is a synthetic fusion protein. So we then ask the question, is there an MLO involved in regulating root hair growth? And what we did is we looked at some single cell data publicly available, and we found that MLO15 is really specific to root hairs. We can zoom in right here. So green is high expression here. And when we look at reporters, we can see strong expression of MLO15 at the tips of root hairs. And if we get an MLO15 mutant, we see dwarfed root hairs, a phenotype that's similar to Feronia, but a little bit more. So next we looked at root hair growth in Colombia versus MLO15. So first, number of root hairs doesn't really change, but we see final length is much shorter. And if we just measure root hairs along a root from the tip up to the hypocaudal, we can get a faux growth curve. And we can see pink here is Colombia, blue is MLO15, so it grows less. And our MLO15 GFP can complement and maybe even get longer root hairs. Okay, and we can also show that MLO15 root hairs shown here in blue grow at roughly half the rate of wild type, and they're very similar to Feronia mutant root hairs that are still maintaining growth and haven't burst yet. Okay, run out of time. So question is, does MLO15 subcellular localization change during different stages of root hair growth? Like we see with Nordia. So here's some light sheet with MLO15 GFP. We can see it's very tip localized. If we look more closely, we can see it's not there yet during root hair initiation. And then it comes on once the bulge starts to be made. And then it seems to be being trafficked down to the tip. We also see if we look at old root hairs that have already stopped growing, it seems to be that the GFP signal gets sequestered, maybe in the Golgi. So the idea is that once the growth is no longer needed, you don't need the MLO to be there at the tip, so then it gets endocytosed. Okay, so last question, does MLO15 also affect calcium dynamics in root hairs? So we did the same experiment. We used Colombia, MLO15, and then a 35S MLO15 construct that complemented the MLO15 mutant phenotype. And just roughly at first, we could see, okay, the MLO15 root hairs seem to have dimmer signal, but this is not a ratiometric. So we looked also at patterns. And in wild type, we see a frequency that's at this about 0.04 in Colombia. So that's every 25 seconds, there's a peak. In 35S-MLO15, this gets completely deregulated and we've got peaks all over the place, different frequencies. And then we recover it back to almost the same frequency and express it. From this, we made a model that somehow during root hair tip growth, again, we have Feronia acting to control what's happening and maintaining tip integrity. Probably there's other calcium channels also involved in setting up calcium gradients. It's been shown several CNGC channels are involved. And then maybe the MLOs, again, are acting to boost that signal. And then we've also got some data I'm not going to talk about, but that we can also recover reactive oxygen species accumulation in root hairs by just adding in FA Nordia construct. Okay, so to conclude, we have signal-mediated trafficking of the MLO protein Nordia from the Golgi to the filiform apparatus during pollen tube reception. This increases calcium transport into synergids and mediates signaling that leads to proper communication and pollen tube rupture to release the sperm cells. Feronia signaling is required for Nordia trafficking, but FA Nordia can bypass Feronia in both synergids and in roots. And finally, we've discovered a new function for an MLO15 in tip growth in root hairs. So I just want to leave you with a couple of thoughts here. Got a couple of minutes, I think. So what we think is maybe these MLOs are kind of acting like the volume knob on an old-fashioned radio, right? So you set your AM or FM band, and then you tune into a station, right? So that's maybe what Feronia is doing. It's helping to tune it in. And then we have these MLOs that are then able to turn up the volume so that you get the proper response happening. Why would you want to do this, right? So especially in pollination and pollen tube reception, you want to make sure that if you've got the right pollen tube, you want to really ensure that it's going to burst. And so synergids, before a pollen tube arrives, they're lure factories, so they're pumping out these attractants. And then when the pollen tube arrives, they need to stop attracting and they need to switch to reception mode. And so you could imagine if you don't have the right ligands, the Ralphs that are going to trigger Feronia, then the ovule doesn't want to waste time on that pollen tube. However, if it has the right ligands, it wants to make sure that that pollen tube will deliver its sperm cells. So I think that's part of this signal-mediated trafficking. So let's keep it at a level where we can kind of trigger it, but then once it gets triggered, we really want to make sure it happens. And maybe in the interspecific crosses, what's happening that's causing pollen tube overgrowth is that there's just not quite enough triggering, and it doesn't reach the threshold that then the FA Nordia can do its job. Okay. Root hairs, similar deal, right? You want to keep a balance. Too much calcium is as bad as too little. And maybe modulating these MLOs helps to sort of keep things moving in the right direction. All right. I want to leave you with one more thought. So there are actually 17 Feronia-like genes in Arabidopsis. So most species, these are big gene families. There's 15 MLOs. Lots of these now have functions assigned to them. MLOs were working on filling out what functions are for all of these. But I think it's interesting to think about, especially with plant reproduction. So far, 7 Feronia-like genes have been implicated in places like the stigma, pollen tube tip growth, in the transmitting tract, and at pollen tube reception. And 4 MLOs have been implicated on the pollen tube side, so with pollen tube tip growth and pollen tube reception. So we also think there's more MLOs involved places. So I think this is a really interesting conservation. And here I list some of the genes that have been found. And I think if you're thinking about any process where cells are talking to their external environment, often there's calcium involved in this. And I think having this ability to work on the volume knob maybe has great advantages for plant cells. Okay, so with that, I'd like to thank the main people who did this work. So Jing and Yan did the early work with the live imaging of pollen tube reception. Yan did all the FA Nordia and synergids. And then Sienna did all the root hair stuff. So we had never worked on roots in my lab. She figured it out. She went to Chris Steiger's lab, learned how to use the light sheet and calcium stuff. And she's finishing her PhD this spring and was slow to look for postdocs. So if anybody wants a really great postdoc, Sienna's your girl, let me know. And then Somia's a new student in the lab that started a year ago, and she's working on Nordia trafficking and trying to understand how that's working within synergids. And I'd also like to acknowledge the National Science Foundation. And with that, I can take any questions.
I'm wondering if you're willing to share how your thinking is emerging on the functional need for oscillations and the mechanistic basis for oscillation.
So I was just talking about this with Maria. I don't know, like, is it we said, is it a red herring, right? How do we, is it really needed? At least in pollen tube reception, if you damp down those oscillations, the pollen tube doesn't burst. So we do know something's going on, but like I said, we're now starting to lean more to it's what's happening outside the cell than what's inside. Yeah, I don't think the field really knows. But we do know in Feronia, we do see these really aberrant oscillations, which is very bad for the root hair, or correlates with bad outcomes for root hairs. Yet another.
Do the oscillations function like a thermostat, that the oscillation itself isn't so important, but you're trying to get a certain amplitude?
Possible. So, so the question is, do the oscillations function like a thermostat? And Mark Johnson's lab at Brown University, they did a bunch of interspecific crosses, looking at synergids and oscillation patterns. So some of them, the pollen tubes could make it and then they don't burst. They didn't see any correlation between frequency or amplitude and the ability to receive the pollen tube. So we know they need to happen, but I don't know, it was kind of disappointing. We all thought, oh, this is going to be the thing. But yeah, it's good mystery. Joss.
Hey, Sharon, that was a great presentation. So thinking about the bursting mechanism, I can imagine you could be that you get perturbed cell wall deposition, growth, and so target pressure blows it open, or it could be cell wall degrading enzymes from the itself, or both. Do you have any idea as to how that operates?
No. So, I mean, people think that, so there's a similar Feronia-like Oral K, Ralph stuff. There's autocrine, paracrine signaling. And so people think, okay, that gets disrupted. But what's weird to me is the pollen tube drives up, stops, and then there's communication, and then it doesn't just burst, it grows again and then bursts. And so they're still intact enough to allow that last growth phase. I'd like to hear your opinions as a cell wall person. But yeah, I mean, it could be that degrading enzymes are activated somehow. Yeah.
Building from that, then what happens to the filiform membrane? Is this burst open too?
So the filiform apparatus, the Grosnickhouse lab has showed, so it's an invaginated membrane, and it's kind of, as the pollen tube grows in, it expands like a glove, like a balloon, right? Or like sticking your hand in a glove. So part of it could be you need to change calcium there so that it actually can loosen somehow. And there's little bits of cell wall there. It's not completely covered with cell wall. Yeah, old literature said it entered the synergid and then burst. That doesn't seem to be true.
Has anybody looked at pH dynamics? Because if you've got a drop in pH in a very localized way, then expansins suddenly are active, and that's it. They go.
I'm not aware of anyone doing that. We tried some in vitro
Yeah.
stuff, but we need to put the extracellular pH.
That could be a way of achieving extremely localized, very rapid
Yeah.
wall loosening at that spot.
It's good begin.
I just wonder about calcium in that vicinity. So calcium comes with the cell wall, right? Right. So I kind of wonder whether calcium dynamic at the tip in depletion from the cell wall can contribute to the burst or activity of the enzymes, or yeah.
So it's, I mean, one hypothesis is pectin cross-linking. There's a lot of pectin in polytube cell walls. So maybe you're depleting, I don't know if it would be enough to deplete and then cause changes in cell wall, for sure. That might be part of it.
Any ideas about how MLOs got co-opted for pathogenesis?
Yeah,
What you're saying?
yeah.
Okay, so how would that happen? They're modulators?
So yeah, so that's a good, right, because it's not like a resistance gene, it's a susceptibility factor, right? And so if MLOs were there during monitoring of cell integrity and things, right? So you have Veronias, Veronia-related monitoring cell wall integrity, then bringing in a backup MLO to turn up the volume, then maybe somehow that you could imagine that somehow crossed over to pathogens being able to take advantage.
It's pretty amazing how FA Nordia complements most of the Feronia defects or phenotypes. Did you see any negative trade-offs of dirty?
Yeah. So Margaret's question is, are there negative trade-offs of overexpressing this FA Nordia? Plants look completely normal. We've done all kinds of measurements. Rosette size is the same. Everything looks fine. But they do have differential responses to stress. So salt and drought. We're collaborating with a physiologist in our department to do a bunch of water relation stuff. So maybe the plants are primed to be able to respond to certain stresses. Yeah, I was shocked. I was like, this is going to mess up the plant. We're deregulating calcium and it doesn't. So it just shows you that there's a lot of compensation that can occur.
On the stress topic, in your slide you mentioned that Feronia somehow modulates heavy metal stress. And so I kind of wonder with regard to root hair growth. So iron deficiency causes root hair growth. And so I kind of wonder what your thoughts about, like, is Feronia MLOs in wall, like anything known? Because I haven't heard
we've not tested it, but we could, or yeah. I mean, I think Feronia is a weird one. So basically, labs have done transcriptomic stuff, pull downs. Any interactor you look at, you can find a phenotype that fits what you want to see. So definitely there's some stuff with translation and messenger RNA processing. I don't think that stuff is going to be complemented with an MLO with that being pretty but yeah, there's some stuff in the literature I'm not sure about, but it's very easy for us to test. We could test that. Oh, one more thing, I also have an open postdoc position for at least a year. If anyone's interested, please let me know.
Could you check online if there are any questions over there?
No questions.
Thanks, Sharon.
This has been a production of Cornell University, on the web at cornell.edu.